Also, thank you we want to thank the hosts for letting us give this talk. So Lisa and I are going today talk about our recent manuscript about microbial enrichment and storage for metagenomics of vaginal, skin, and saliva samples. So we're going to talk about our recent paper, so you can look it up after or during our talk in iScience by Cell Press. But let's first take a step back because I want to tell you about the Isala project. The Isala project is a large-scale citizen science project on women's health that was established by the lab of Professor Sarah Lebeer. It was inspired by a role model, Isala van Dis, who you can see here, was the first female physician in Belgium, but also a feminist and an activist. We have lots of scientific goals on this project, so we aim to map the female microbiome in Belgium and study its influencing factors, but also set up a large biobank to study beneficial vaginal microorganisms. And next to scientific goals, we also have lots of societal goals. So we want to break the taboo on vaginal health with our online community and participants, make the topic open for discussion, also empower women to take their own vaginal samples. And this one is actually quite important for the following presentation, and also bring women in science more into the picture. So we have a few flows. I'm not going to talk about our cross-sectional flow a bit, but I'm proud of showing you a little bit about it. We are working on the manuscript now and preparing it for submission. This included more than 3,300 participants that donated 2 vaginal swabs on one time point and filled also out a survey. So for this study, we had to prepare and decide which swab we would use, which collection method. And for the longitudinal flow, of which we are actually now sequencing the samples, and that's why the coming slides will be more clear. We included almost 300 participants that were going to sample over 2 menstrual cycles on 6 time points. So we really wanted that our citizen science project, that the participants felt safe. So we invited them to self-sample in the privacy of their own homes, in their own bedrooms. They then stored the samples in their own fridge for 3 weeks accordingly, and then send them over to a lab on room temperature. So after each cycle, they sampled their vagina, skin, and saliva, and then also filled out some surveys. But as most of you know, for large-scale citizen science project, but also microbiome projects, there are a few bottlenecks. The two that we really defined were the storage conditions and the DNA extraction methods, because we, as I said, it was self-sampling, and the study was actually launched in 2020. So during the pandemic, we had to figure out a way that our samples were reliable and the results as well. So our first research question is, what is the effect of longer storage and different stabilizing buffers on the microbiome profiles? And as I said before, we were going to study the vagina, skin, and saliva. And it's known that these 3 body sites have high amounts of human reads. And with the study, we also actually want to shotgun sequence, but minimize the cost a bit on sequencing. So we wanted to deplete the human reads as much as possible without disturbing the profiles. So another research question for this project was, what is the effect of different host DNA depletion methods on the microbial profiling and human reads? And as you know, for each project, there's a long experimental flow, and here you see a summarized version of it. I'm going to focus on the self-sampling kit. As you can see, we put lots of effort in the aesthetics, but also on the scientific content. So deciding on which swab the thousands of participants were going to use, we compared the eNAT and the M swab from Copan. And here we specifically looked at our study design. So 3 weeks on room temperature at the participant's home, and then we mimicked it as well as if the samples would be on the way for 3 days on room temperature using the National Postal Service. And then we also looked whether or not we should be immediately processing our samples, or whether we could wait till all the samples came in and then process them all together, because that also facilitates the lab work. And here we found actually that the eNAT swab from Copan preserved the microbial profiles the best for the studied storage time, and that was very relevant for the Isala project. So here you actually see the 16S profiles of vaginal microbiome over the 4 time points. And you can see for the eNAT swab, they barely change. But compared to the M swabs, where the M swabs you see an increase and decrease of very relevant vaginal taxa. So it was very clear for us that we would be using the eNAT swab in this aspect. And also we advise other people who work on microbiome projects, especially with vaginal samples, to use the eNAT swab.
Okay, and after the arrival and storage of the swabs, we went over to DNA extraction and sequencing. For the sequencing, we wanted to use both 16S and shotgun metagenomic sequencing, because 16S gave us an overall bacterial community composition for all the samples. But for selection of the samples, we also want to do shotgun to be able to do functional analysis as well. We knew already that swabs contain a high amount of human epithelial cells, and that this can result in high proportions of human reads compared to the bacterial reads. And this then can lead to a decreased sensitivity in microbial detection and in higher costs. And therefore, we looked into different methods to deplete the host DNA before extraction for the Isala project. As a golden standard, we used the power-fecal DNA kit because this was used before a lot in our lab and in literature. Then a first method that we analyzed is an experimental method based on the osmotic lysis of mammalian cells and treatment with PMA to degrade the human DNA. And then in addition, we also tested 2 commercially available kits that use host depletion based on the lysis of eukaryotic cells and an enzymatic degradation of host DNA. To decide which kit to use in our Isala project, we did analysis such as qPCR and sequencing. Based on qPCR, we selected that the host0 kit performed the best in depleting DNA. You can find this data in our paper. But just DNA concentration didn't tell us anything about the effect on the microbial profiles. And therefore, we did 16S sequencing on samples by using the 4 different methods. The dendrogram visualizes the clustering of different samples. And we expected that all samples would cluster according to their niche and not according to the method. But for the PMA method, we saw that the clustering was mostly by method. And so this suggests that the technical effects of this method overruled the biological differences between the vaginal skin and saliva samples. And this was also seen in the Bicurtus dissimilarity, where we see higher dissimilarities for the PMA treatments. After the 16S sequencing, we selected the host0 method to continue with because this was the most time efficient and did give the lowest cost. And based on 16S differences in beta diversity, etc., there was no significant difference of this kit with the QIAamp. And when using this method, you can see that the blue dots gave a significant increase in bacterial proportion of our reads. For example, for the vaginal samples, we did have 96% of human reads without the host DNA depletion and only 13.6% with host DNA depletion. Because 16S already told us that there might be a taxonomic bias introduced by using the host DNA depletion methods, we also looked into this bias in our shotgun data. And we detected a taxonomic bias that was dependent on the biological differences between the body sites. We saw a reduced relative abundance of gram-negative taxa within mainly skin and saliva samples. For example, for saliva samples, you can see a reduced relative abundance of Neisseria, Prevotella, and Haemophilus species over here when using the host0 kit. This taxonomic bias was less pronounced in the vaginal microbial communities because healthy vaginal communities are mainly dominated by gram-positive bacteria. And then to conclude our study, we saw that the eNAT lysis buffer can keep the microbial composition stable during various storage conditions. We also saw that the host 0 microbial DNA kit performed the best in human DNA depletion for shotgun metagenomics of all the kits that we tested. And then the host DNA depletion introduces a larger bias towards gram-negative taxa, therefore we do not recommend it for skin and saliva samples. But we suggest a body site-specific approach based on the microbial composition to minimize this taxonomic bias. So we should suggest a host DNA depletion method for vaginal samples. To end, I would like to thank all the co-authors of this publication and the whole team of the Laboratory of Applied Microbiology and Biotechnology of the University of Antwerp. And thank you for your attention.